When investigators get to a crime scene, sometimes there are stains at the scene of the crime that they have to determine some information about. So if you get to the scene of a crime and there's an unknown stain, say it's a red stain that they find in the kitchen, investigators will go through a series of questions to kind of figure out what that stain is to the best of their ability. The first question is the stain really blood? Because it could be something else, right? It could be Kool-Aid, it could be food coloring, it could be paint. So that's the first question is the stain really blood? If the stain is blood, is it human blood or animal blood? Because if it's animal blood, they're probably not going to do as much investigating. And number 3, if the stain is human blood, what is the blood type? So today we're going to talk about the tests that are completed in the field and in the lab that help investigators answer these questions. So the first question we're going to focus on is our unknown stain really blood or not? There's 3 possible tests that they can run. 2 tests, the first 2 tests are for stains that you can visibly see, and the third test are for areas that they think a crime could have occurred in but would have been cleaned up. So the first test is called the Kastle-Meyer test. It's a mixture, the Kastle-Meyer solution is a mixture of phenolphthalein and hydrogen peroxide. And hemoglobin, which is found in your blood, will react with phenolphthalein because it's basic and will cause a deep pink color if blood is present. So how investigators do the test is they usually take a Q-tip and they swab the stain that's in question, and then they take the Kastle-Meyer solution and they add a drop or 2 to the Q-tip, and if bright pink forms on the tip of the Q-tip, then they know that their stain is in fact blood. You see this a lot of times on all the TV shows that they do. The second test that you can do if you can see a stain is what they call the Hematest tablet. It's very similar. It's a color change test. The tablet will react with the heme group in blood and it causes a blue-green color change. Oftentimes investigators will use the Kastle-Meyer solution more frequently because it's a cheaper solution to make and use. The third test to determine if a stain is blood or not is called the luminol test, and this one is a little bit unique in itself. The luminol test is used when investigators think there could have been blood in an area, but they think that it's been cleaned up. So for example, say you had a suspect at a very bloody murder. They get a search warrant for their house and they think that the person had went into the bathroom and cleaned up after they had committed this crime. Okay, but then they had cleaned the whole bathroom up so you couldn't see blood, but they still thought that it was there. They would use luminol. Luminol is a chemical that the smallest amount of blood present will still react with the luminol, and it produces light. So the picture in the bottom right-hand corner here is a bathtub that was completely cleaned up. Investigators sprayed luminol all over the tub, and they turned the lights off, and anywhere that there was glowing light, that was where blood was present at some time. So it only needs the smallest molecules of blood to react with the luminol and it will produce light. So how this test works is investigators will take the area that's in question, they'll turn the lights off, and then they'll spray a solution of luminol all over the area, and anywhere that's glowing, there will have been blood present there at some point in time. So those are the 3 tests that you can do to determine if a stain is blood or if it's some other substance. The next question that investigators have to answer is if a stain is blood, is the blood human or animal? And there's 2 tests that you can use for that as well. The first test is in the lab, so if they can take a little sample of the blood back to the lab, they can do microscope observations. Animal blood is usually large nucleated cells, whereas human blood is smaller cells and there's no nucleus because they're mostly red blood cells. And so you can see in the pictures here, this is horse blood, this is frog blood, this is human. So it's noticeably different for them to view under the microscope. But if they're in the field and they need a quick, hey, is this human blood or animal blood, and they don't have time to wait for it to go to the lab and get microscopically analyzed, they'll do what's called a precipitin test. So how the precipitin test works, it's kind of a process. You have to have what's called a human antiserum, and the human antiserum is then used in the field to drop on your sample and if there's agglutination or clumping, then you know human blood is present. The way that they get the human antiserum is kind of interesting. Excuse me. Blood is injected, human blood, is injected into a rabbit. The rabbit forms antibodies against it because it's considered foreign for the rabbit, right? So the rabbit's going to make antibodies against it. Then the rabbit's blood is extracted out of the rabbit. So they take like kind of a little tube of the rabbit's blood out, and that is the human antiserum. So the human antiserum is basically rabbit's blood that has antibodies in it. They then have this kind of in a dropper bottle, and they have it in a kit. And so if they're out at a crime scene and they need to figure out if it's human or animal blood, they'll take a little bit of the human antiserum and they'll drop it on the blood in question. And if the blood clumps up, then they know it's human. If it doesn't clump, then it's some type of animal blood. And they don't know what kind of animal, they just know it's animal. It's a very sensitive test. It only requires a small amount of blood for it to happen. So when you're doing a precipitant test in the field, you're looking for agglutination or clumping with the human antiserum, then you know that human blood is present. The last test to do is if the blood is human, the last test that investigators will run is a blood typing test. To run a blood typing test, you need a blood typing tray similar to this. Okay? You're going to use antiserums. You have an A antiserum, a B antiserum, and an RH antiserum. If your blood is put into an antiserum and it clumps or it agglutinates, you know that that blood type is present. So clumping is a positive for that blood type. No clumping equals a negative. So for example, if we were looking up here, okay, and this is our A antiserum, and we put blood in it and there was no clumping that happened. Then in our B antiserum, we put blood in and it did clump. We know that we have B blood. And if we put our blood in the RH antiserum and it does clump, then we know our blood is positive, not negative. Okay, so if there's clumping, it means that blood type is present. If there's no clumping, it means that that blood type is not present. So they would do a blood typing test. They will do a blood typing test at the lab, and that would determine what the blood type of the unknown stain is. And then they can use that information to take a look at their suspect pool, to eliminate suspects that they know can't match the blood type that was there, so on and so forth.